If you want to study a complex, beautiful ecosystem like a coral reef, you don't just scoop the whole thing up, drop it into an industrial-sized blender, and hit puree.
Right. No.
Because, you wouldn't learn how the clownfish interact with the anemones or where the sharks hunt by looking at a completely blended soup.
Yeah, that completely destroys the geography.
Exactly.
Yeah.
Yet, for decades, that is essentially how we have studied the genetics of cancer. We take a tumor, we grind it up, and we sequence the RNA.
We make a smoothie.
Right. Welcome to this deep dive. Today, we are looking at a 2021 study published in Nature Communications by Anderson and colleagues that, well, it completely changes this paradigm.
It really does. It's groundbreaking.
Our mission today is to understand a technology that maps the tumor ecosystem without the blender. And we're focusing specifically on 36 tissue sections taken from 8 individuals. These are identified as patients A through H who all had HER2 positive breast cancer.
And it is a critical study to unpack because HER2 positive breast cancer makes up roughly 15 to 20% of all breast cancers.
Which is a lot.
It is. And historically, it's a highly aggressive subtype. Now, we have developed revolutionary therapies that target the HER2 receptor, but we still see patients who inexplicably experience resistance to these treatments.
And the answer to why that resistance happens?
Well, it often lies hidden right in the tumor's microenvironment. Specifically, how the immune system is physically interacting with the cancer. But as you mentioned with your blender analogy, traditional sequencing destroys the exact geographic context we need to actually see those interactions.
Right, which brings us to the core of this paper, Spatial Transcriptomics, or ST. So if traditional single-cell RNA sequencing is our fruit smoothie, where we know we have exactly 20% strawberry and 10% kiwi in the cup.
But no idea how they were arranged.
Exactly. Then spatial transcriptomics is a fruit tart. You get the biological data, the gene expression, but you also see that the slice of kiwi is sitting right next to the strawberry.
I love that analogy. It's a perfect visual.
Thanks. But I do want to push back on the resolution here before we get too excited, because for you listening, how detailed is this map, really? Are we looking at biological continents, or are we zooming all the way down to individual cellular houses?
So to answer that, we have to look closely at the mechanism of the microarray itself. The researchers use a tiny glass slide, and it's printed with a microscopic grid of spots.
Okay, spots, like polka dots.
Sort of, but microscopic. Each individual spot is exactly 100 micrometers wide, with a 200 micrometer center-to-center distance between them.
Got it.
And inside each of those 100 micron spots are millions of tiny capture probes. The crucial detail here is that every probe within a specific spot contains a unique DNA barcode.
Like a zip code.
Yes, you can think of it exactly as a molecular zip code.
So they take a frozen slice of the breast cancer tissue, and the paper mentions is just 16 micrometers thick, which is incredibly thin, and they lay it flat over this grid.
Precisely. They lay the tissue down, and then they apply permeabilizing chemicals. This, it gently pokes microscopic holes in the cell membranes.
Like poking holes in a water balloon.
Right.
Yeah.
And the RNA from the tissue physically falls down onto the slide and binds to those capture probes. So the researchers are permanently tagging the gene expression with a fixed set of GPS coordinates.
Wow.
But to address your earlier question about resolution, a 100 micron spot is not a single house. Depending on the density of the tissue, one spot contains anywhere from 0 to about 200 cells.
Okay, so it's more like a neighborhood.
Exactly.
We are mapping out zip codes, not individual mailboxes, so that transcripts of dozens of cells might be mixed together, but it is a highly localized, geographically fixed neighborhood.
Yes, and the first thing the researchers did with this new map was try to identify distinct territories. This is where they actually pitted artificial intelligence against the human eye.
I found this comparison fascinating. So, okay, let's do a quick recap here. We're using barcoded arrays to get the geographic coordinates of gene expression. So if you had a GPS map of a tumor, what specific biological battlefronts would you want to zoom in on first?
Well, the researchers wanted to see the boundaries, but they didn't just look at the raw map and guess where those boundaries were. They used the underlying data to group identical neighborhoods across the tissue, completely blind to what the actual tissue looked like visually.
Right, they used expression-based clustering.
Yes, specifically a shared nearest neighbor approach using an algorithm called SIROT. Now, high-dimensional RNA data is incredibly complex. I mean, we're talking about thousands of genes being expressed at different levels all at once.
It's just a massive wall of numbers.
Exactly. So the the Seurat algorithm calculates the mathematical distance between the transcriptomic profiles of every single spot. It groups them into clusters based entirely on shared gene expression without ever actually seeing the physical structure of the cells.
Meanwhile, they had a trained pathologist look at the exact same tissue the old-fashioned way.
Right.
The pathologist used a standard hematoxylin and eosin stain under a microscope, visually hunting for the physical structures of, like in situ cancer, invasive cancer, or immune infiltrates.
And at a macro level, the algorithm and the pathologist largely agreed. The mathematical clusters mapped beautifully onto the pathologist's visual annotation.
Which is comforting.
Very, but the purely data-driven approach uncovered crucial hidden layers that the human eye simply cannot perceive.
Let's look at patient E to ground this, because this case study blew my mind.
Oh, patient E is a perfect example.
The pathologist looked at patient E's tissue and saw 2 tumor foci, 2 distinct clusters of cancer cells that were physically separated but visually identical. Based on their morphology, they looked like the exact same type of cancer.
But the spatial transcriptomics clustering proved they were entirely different at a molecular level.
It really makes me think of looking at 2 identical houses on the same street. From the outside, the architecture is exactly the same. But if you point a thermal camera at them, you realize one is secretly an icebox and the other is a blazing sauna.
That thermal camera analogy perfectly captures the concept of intra-patient heterogeneity. In patient E, the algorithm identified one of those visually identical clusters, labeled PE3, as highly enriched for immune response pathways.
So it was biologically hot.
Exactly. It was buzzing with humoral immune activity, meaning antibody-driven responses. But the other cluster, PEC4, was dominated by cell death and apoptotic pathways, meaning the cells were actively triggering their own programmed death.
Essentially hitting the self-destruct button.
Right. And then there's patient H.
Oh, patient H, where the algorithm found heavy immune clusters hiding deep inside an area that the pathologist had simply labeled as in situ cancer.
The human eye just couldn't see the immune cells hiding in the midst because the physical structure of the cancer cells dominated the visual field.
But the gene expression doesn't lie.
It doesn't. It picks up the molecular signatures of those infiltrating immune cells even when they are visually obscured, which means, practically speaking, 2 tumors that look identical under a microscope might require entirely different treatments because their molecular neighborhoods are fundamentally operating under completely different rules.
Okay, we are about 5 minutes in, so let's just do a quick milestone recap to cement what we've established so far before we move into the more complex math.
Good idea.
Bullet 1, we aren't just looking at random RNA soup anymore. Spatial transcriptomics gives us a physical map of gene expression using 100-micron spots.
Right.
And bullet 2, by using gene-based clustering, Seurat reveals hidden identities or heterogeneity in tumors that the human eye completely misses.
Spot on.
But this raises a massive logistical problem that I'm actually still stuck on.
Okay, what's that?
If every 100-micron spot holds up to 200 cells, and we see a spot glowing with both a cancer signature and an immune signature, how on earth do we know who is who? Because it feels like we've just made a smaller, neighborhood-sized smoothie.
That is a great point, and that was actually the major analytical hurdle of the study. Because the spots lack single-cell resolution, the researchers had to unmix the signals computationally.
Unmix them?
Yes. They utilized a deconvolution method called stereoscope.
Stereoscope. Okay, walk me through exactly how a computational model unmixes a physical biological sample.
So stereoscope relies on probabilistic modeling, and crucially, it requires an external reference data set. The researchers took a completely separate traditional single-cell RNA sequencing data set from 5 other HER2 positive tumors.
Okay, so traditional smoothie data.
Right, but remember, single-cell data has perfect one-to-one cellular resolution, just zero spatial context. It tells you exactly what the pure RNA profile of a single macrophage or a single T cell looks like.
Ah, so stereoscope takes the precise blueprint of a pure cell from the single-cell data, holds it up against our messy 200-cell spatial spot, and uses probability to guess the exact ratio. But doesn't that leave a massive margin for mathematical error?
It would if it were just a simple linear guess. But stereoscope is highly sophisticated. It models the gene expression of the spatial spot as a combination of the expression profiles from the single-cell reference, but it accounts for statistical noise and variance.
Okay.
It mathematically iterates over and over until it finds the most probable combination of cells that would naturally produce the exact RNA mixture seen in that specific spot. It essentially calculates the exact recipe.
Like this spot is 20% macrophages, 50% cancer cells, and 30% T cells.
Exactly. And they didn't just stop at broad categories. The paper notes they categorized the reference cells into highly specific tiers.
Right, I noticed that.
They mapped major tiers, like general T cells, but then drilled down to minor tiers, like CD8 positive T cells, and even further into subset tiers, isolating T cells that were actively expressing specific interferon response genes.
So they mathematically reconstructed the entire ecosystem, cell by cell, without ever physically separating the tissue.
Yeah.
Which means we now have a map of exactly who lives in every single neighborhood. And in what exact proportions. But knowing who lives on the block doesn't necessarily tell us who is talking to whom.
That's the next step.
Right, so let's recap this subtopic for the listener. Stereoscope acts as a mathematical unmixer, calculating the exact recipe of cell types inside every spot on our map. So the big question for you now is, now that we know who lives in every neighborhood, how do we figure out who is talking to whom?
To figure that out, they turned to Pearson correlations.
Okay.
They systematically looked at the deconvolved cell proportions across all the thousands of spots and statistically asked the data, hey, when the proportion of cell type A goes up in a specific zip code, does the proportion of cell type B consistently go up as well?
Oh, I see.
If two specific cell subsets are consistently found co-occupying the same 100-micron spots across the entire tumor, it strongly implies a functional colocalization. They're hanging out together for a reason.
And when they ran this correlation across the entire data set, they found a massive, undeniable connection between a very specific macrophage state, which was labeled M02CXCL10, and a very specific T cell state, labeled T cells IFI-T1.
This specific interaction is arguably the most critical discovery in the entire paper.
Really?
Yes. And to understand why these two cells are constantly finding each other, we have to look closely at the genes they are actively expressing.
Let me offer an analogy here for the audience because the mechanism is just fascinating. We shouldn't think of this as cells just bumping into each other by random chance. It's more like an encrypted radio distress signal.
Oh, I like that.
The macrophage is broadcasting a highly specific chemical text message into the microenvironment. Specifically, the chemokines, CXCL9 and CXCL10.
Right.
This signal is effectively encrypted because it can't just be intercepted by any random cell. It is specifically meant to pull in T cells that carry the exact corresponding antenna, which is the receptor CXCR3. They're inviting them to an interferon party.
That is an excellent way to visualize the chemokine gradient. The macrophage releases CXCL9 and CXCL10, and the T cells equipped with the CXCR3 receptor follow that chemical gradient through the physical tissue right to the macrophage's exact location.
But why are they doing this? What is the purpose of this encrypted meetup?
The researchers discovered that this extreme colocalization happens almost exclusively in spatial regions that exhibit a high type I interferon response.
Okay, break that down for us.
Type I interferons are vital signaling proteins. You can think of them as the immune system's primary alarm bells. In a cancer microenvironment, this alarm can directly inhibit the proliferation of tumor cells or activate aggressive anti-tumor immunity.
The paper points out something highly specific about this relationship, though. It's asymmetric.
Yes. The asymmetry is a profound detail. It means you can have the type I interferon alarm ringing in a tumor neighborhood without the T cells and macrophages being present.
Oh, really?
Yeah. Other structural cells or immune variants can trigger or respond to interferons. However, whenever those two specific cells, the broadcasting macrophage and the receiving T cell meet up, the interferon alarm is almost always ringing at maximum volume.
So they're either deeply drawn to the most intense alarm zones, or their physical interaction is the very mechanism amplifying that alarm in the first place.
Exactly.
Okay, we hit another 5-minute mark, so let's pause and lock in the logic here with our milestone recap. Bullet 1, we deconvolve the spots using stereoscope to find exact cell proportions.
Yes.
And bullet 2, we discovered a highly targeted interaction where macrophages use CXCL chemokines to physically recruit T cells to regions with high type I interferon activity. We're basically watching the immune system coordinate an attack in real time.
And that coordination scales up, because if T cells and macrophages are coordinating locally, the researchers wanted to know if the immune system was building larger infrastructure.
This is where it gets crazy.
It really does. This led them to investigate tertiary lymphoid-like structures, or TLS.
Let's define what a TLS actually is, because this feels like the absolute holy grail of the paper.
It is!
Normally, your immune cells get trained and presented with threats in your actual lymph nodes, which are, fixed organs located throughout your body. You have them in your neck, your armpits.
Correct. But in the presence of severe chronic inflammation or highly immunogenic cancer, the immune system will sometimes completely bypass the standard lymph nodes. It constructs a pop-up, forward-operating military base directly inside the tumor tissue itself.
They literally build a pop-up camp right on the front lines to train the troops.
Exactly. A tertiary lymphoid structure exists to present tumor antigens to T cells right where the fight is happening, drastically cutting down response time.
That is incredible.
Clinically speaking, their presence is one of the most massive predictors of whether a patient will survive or respond favorably to immunotherapy. If your body is actively building TLS in your tumor, your prognosis generally improves significantly.
But wait, think about this. If spatial transcriptomics isn't single cell, and a TLS is a highly organized physical architectural structure made of thousands of cells, how could the researchers possibly see them? A 100-micron spot might capture a piece of a TLS, but it can't show you the architecture.
You're right, they couldn't see the visual architecture, but because of the stereoscope deconvolution, they could see the extreme concentrated colocalization of B cells and T cells.
The primary cellular building blocks of a TLS.
Exactly. They identified spots where the algorithm flagged massive, overlapping concentrations of both cell types. And based on that, they decided to build a mathematical TLS score to predict where these pop-up camps were located, using nothing but the raw gene expression data.
So they effectively asked the dataset, what is the exact RNA blueprint of a forward operating base?
Yes. They used a linear regression model to isolate the specific genes most strongly associated with these high B-cell and T-cell zones, and they extracted a very specific 171 gene signature.
And the genes that top that list are not just random markers, they are the actual functional machinery of the immune system. Let's look at why a few of these matter, because it's so cool. We see MS4A1, which encodes for CD20.
Right, that is a calcium channel critical for B-cell activation.
And we see B2M.
B2M is essential. It stabilizes the MHC class I molecules.
Wait, what does that mean in plain English?
If the immune cells are trying to present tumor antigens to each other, B2M is essentially the structural scaffold holding up the molecular ID badge so the T cells can actually read it.
Oh, that makes perfect sense. And then TRBC2, which is literally a structural component of the T-cell receptor itself. Seeing the actual biological machinery top the algorithm's list proves the math is genuinely finding these lymphoid structures.
It does.
But, identifying pattern in your own tightly controlled data set is one thing. The standard of proofs in science requires validation.
And they provided two distinct levels of validation. Level 1 was physical in tissue validation.
How did they do that?
They took extra tissue sections from patient H. These were sections sliced directly adjacent to the ones used for the spatial sequencing, and they applied traditional immunohistochemistry, or IHC staining.
So back to visual microscopy.
Yes. They utilized antibodies specifically engineered to bind to CD20 on B cells and CD3 on T cells.
And when they looked under the microscope, the visual proof was undeniable. Highly compartmentalized clusters of B cells deeply surrounded by T cells. The pop-up camps physically existed exactly where the mathematical map predicted they would be.
It was a perfect match. But level 2 validation is where the true clinical power of the study is revealed.
Okay.
They took this mathematical algorithm, this 171 gene TLS score, trained exclusively on spatial arrays from HER2 positive breast cancer, and they applied it to a completely external data set, the TCGA bulk RNA sequencing data for melanoma.
Whoa, I have to stop you there because that feels like a massive leap.
It sounds crazy, I know.
They took a precise spatial model built on breast cancer and they pointed it at a bulk RNA smoothie from a totally different disease, melanoma.
Yes.
How can you apply a highly localized breast cancer model to bulk tissue data from a skin cancer entirely?
Because it relies on the fundamental universality of the human immune system.
Oh, interesting.
The underlying biology of a tertiary lymphoid structure, the specific genes the B cells and T cells use to build and operate that forward operating base, is highly conserved.
Okay.
It doesn't matter if that camp is being constructed in breast tissue or skin tissue. The architectural blueprint is the exact same. So by applying their TLS score to the bulk melanoma dataset, they were simply looking for the overall volume of that specific 171 gene signature.
And what did the data show?
The model flawlessly identified patients in the melanoma dataset who had high overall TLS scores. And crucially, those specific patients demonstrated significantly better overall survival rates compared to patients with low scores.
Wow.
It perfectly reproduced established clinical findings regarding TLS and survival, but using a totally novel mathematical model.
So what this proves is that the spatial map can be used to forge tools that work even when a doctor doesn't have access to expensive spatial technology.
Precisely.
You can use the spatial transcriptomics chart to build a highly precise compass, and then hand that compass to a clinician who only has a standard biological smoothie.
Exactly. They can run a standard bulk RNA test, look for this 171 gene signature, and know with high probability if their patient is actively building these immune fortresses.
It completely bridges the gap between high-resolution spatial discovery and broad, accessible clinical application. It generalizes across different tissues and entirely different diseases.
It is a phenomenal scientific arc.
It really is. So let's bring all these threads together to fully appreciate what Anderson and colleagues accomplished here. We started with the historical problem of the biological blender, taking a tumor and pureeing it so completely that we lost the geographical map of the disease. Then, by utilizing spatial transcriptomics, we mapped the tumor ecosystem with 100-micron precision. We saw how Surratt's data-driven clustering bypasses the limitations of the human eye, finding hidden molecular battle zones that look perfectly identical under a traditional microscope.
And then we explored how stereoscope probabilistically unmixed those complex neighborhoods. That allowed us to discover that M02 macrophages are actively broadcasting encrypted CXCL chemokines,
pulling T cells through the physical tissue into localized zones with high type I interferon alarms.
Exactly.
And finally, we tracked that cellular coordination all the way up to the architectural level, discovering a 171 gene signature that predicts the existence of tertiary lymphoid-like structures, those pop-up immune bases, which can successfully predict patient survival across entirely different types of cancer.
Which leaves us with a really provocative concept to consider as we look toward the future of oncology.
I love this part. Tell us.
If we can now perfectly map out exactly where, how, and with what specific genes these immune-forward operating bases form, could the future of cancer therapy involve a drug that artificially forces a TLS to pop up?
Oh, wow.
Imagine engineering a targeted therapy that physically constructs one of these immune camps exactly in the weakest, most vulnerable sector of a patient's tumor, literally turning their own microenvironment against the cancer.
Hacking the genetic map to build our own immune fortresses exactly where we need them most.
It is an incredible thought.
It is, and it all comes back to the realization that in biology, just like in real estate, absolutely everything comes down to location, location, location. So for you listening, consider how this changes the way you view the medications or therapies you interact with. Are they just treating the ingredients, or are they actually treating the map? Thank you for joining us on this deep dive. Keep questioning the map, and we will see you next time.